So hello, welcome to Data Carpentry, interaction data with GloBi. So we have a small crowd today on here and online. My name is Kristi Liu. I'm from the UCSB library, and some of you may be thinking, what's someone from the library doing here at a museum conference? I was originally a student at UCSB, and when I was there, I worked with the natural history collections. After I got hired as a staff member, I continued working with the Cheadle Center of Biodiversity and Ecological Restoration as my long-term research engagement. The workshop that we're going to be doing today was originally developed like as our little long-time COVID project, and then after a while, we decided to develop it, or at least start developing it into a carpentry workshop. All right, let me see here. I was going to drop some stuff into the Zoom. Hold on. Where did it go? I'm sorry. Got it? Didn't I screen freeze? No, okay. So let's see here. This is the workshop website. We have our lesson available online if you would like to browse this on your own time. With the general information, carpentries, our disclaimers, so I didn't get anything from anybody. We do have a collaborative meeting note document, which I will share with the Zoom, and also the links to the workshop. Unfortunately, I do not have a moderator, so I'm doing this on my own. So this is our workshop website, and this is the collaborative document that you may ask questions on or put comments on if you're online. For those of you who are in the room, we have the special bonus of I made a game for us to play today to get us into like what are biotic interactions, and how are they useful for your research, and also about GloBi in general. So I'm going to open my slides. So a little bit about me and how I kind of got into this. To start off with, does everybody have their stickies? Who does not? Okay. No? Okay. One person does not have one. But the sticky notes, if you are following in person, we'll use these. If you have a red sticky, that tells me that like you're kind of lost or don't understand what I'm doing. And green sticky means that we're good. And if you're online on Zoom, I'll use the thumbs up and thumbs down icons. So let me go in. And also, we do have a carpentry's code of conduct. So, treat each other politely, respectfully, don't be rude. Pretty much the same as the Spanish and Tadwig conference. So I'm gonna start. So, yay, welcome. So this was a photo taken on iNaturalist by somebody this is a tarantula hawk and dragging off a tarantula. And yeah, we have like a tarantula season in California. So some of us are kind of used to this. Other people are like, oh my God. So I work at the Dream Lab. It's our digital scholarship center, but students don't really know what digital scholarship is. So the occasional financial aid student came in and was like, I need help. And we're like, no, that's not it. So we rebranded and became the Dream Lab. So DREAM stands for Data Research Exploration Access Methods. And this is what we provide assistance with for students and faculty on our campus. If they want to learn more about how to use different data, like research, and also like how to find data for the research, as well as how to use that data. So we handle quantitative and qualitative softwares and data. So we also do a lot of consulting, so ask us anything, and then we also refer out to our HPC. We have a high performance computing center and also a secure compute. So kind of flying in through this, but what is the Carpentries? So we're a worldwide organization of volunteers and also researchers, people in industry, students, faculty, you name it. And we give our time to help teach and also develop lessons in foundational coding skills. So Instructors, me, teaching instructor, and then helpers. I do not have one this time. I thought one would come, but they would assist with answering your questions and also with any general software support, like during a setup, and also lesson development as well. So our main focus is doing a teach-in type. I recognize that like maybe today might not be one of those days, but that's okay. We really are like starting off like very soft entry. So practical foundational skills in Bash are Python, SQL, and OpenRefine. And the backstory of how UCSB joined the Carpentries and started doing this was we were getting comments from faculty that their incoming grad students, especially in the humanities, didn't have the skills that were needed to deal with their data. So we get a lot of students who are maybe like in the humanities who need assistance or at least like help getting started with getting to know how to use their computer. And also the occasional person who comes in to come in for a refresher. So I was talking to somebody and they're like, what is Bash? So who here uses their terminal or their Bash on their computer? Okay, a few people. Who's never heard of this before? It's okay. No, it's okay. Raise your hand. Raise your hand high. It's okay. Okay, so back when the first computers came out, like the little prompt that would come up, that's your Bash terminal. Like it lets you go into your computer. I'll show you a little bit when we go through this workshop. And this is just at UC Santa Barbara. It's our 5th year teaching. I updated our workshop, but during the pandemic, we really like ramped up our teaching. And so far we've had over 1,000 learners since 2019, and we've had 78 workshops as of the last one on July 23rd. So a lot. We offer like a strong pedagogy and we try to use reusable open educational resources and then try to teach those skills to students as well. So this is how I kind of got roped into natural history collection. So some of you may know the Big B project. Our undergrad students did all this modeling here on the side. It's very nice. I just provide the occasional software support. But yes, this is the Big B project. Okay, we're gonna start with this. So who has heard of Karuta? So we're gonna play a little game here as like an icebreaker. I'm sorry for those of you online who might not be able to. So I'm gonna show you a bunch of slides. I have some cards here. We're gonna move this table here. And based on what you think, you're gonna don't fight over the cards. You're gonna choose the card that you think best suits with the interaction on the screen. And this is just to start. So it's a squirrel and a rabbit. What are they doing here? Are they interacting? Are they killing cards with? You get the point. So we're gonna move this table out to the front and I'm gonna have you guys step forward. For those of you who want to play, I have little prizes for people. So to get like some group participation. Thanks. There's a lot of cards, but not all of them are maybe in the thing. Maybe there's too many cards. Yeah, maybe we needed a bigger table, who knows? All right, so I'm gonna go back to my name. For those of you who want to play. All right. Thank you, Yuka. So I'm gonna change the screen right now. So, find the card on the table that best suits what is going on in this picture. And these are all iNaturalist observations I took off iNat. Unfortunately, does not fit, so grab the card that you think best. Yeah. That you think best, and I also have like little keychain prizes over here. So, and then we'll kind of talk about like what ontology relationship works best. And I have a few of these slides, so if we're okay on time, we can keep going. But yeah. Okay, I'm good. Yeah, all right. So, what are some of the cards that you guys picked up? So I'm gonna start picking people. You in the front here.
Provides nutrients for.
Okay, what is another one that we have we can start calling out? Kills, okay.
I have eight. Eight.
Eats, okay. Oh, preyed upon, yeah.
Interacts with.
Yeah, so you can see here, like, the data or what we're thinking about, like, differs per person. But I think the best one here would, the simplest answer here would be eats. So who is that? Yeah.
Congratulations.
Whoa.
A dumb thing made by me. Anyway, so we'll continue on. So there's more.
Wow. Very cool. Wow. Wow.
So what is going on in this picture? And if you're on the Zoom, you can throw in whoops. You can throw your answers into the chat, and I'll try my best to look at them. So let's think about what's going on here. There's a lot of stuff going on here. But what is the interaction best suited, or what biotic interaction would best describe what's going on here? It's a little hard.
Is that a parasite wasp?
I think people are still picking cards, so let's wait a little bit. But yeah, there's in the back. No? You're good? Okay. Okay, so let's talk about what cards we picked. So what are some ways to describe the interaction in this image?
Has habitat.
Okay. Maybe. Killed by, perhaps. Did you pick a card, Miwa?
Also, I distribute count, but the stack I had also had ladies and things, unless it's also in here.
Okay, like I think there's one that about like making houses in somewhere.
Is it where your home stuff? Is it where your home stuff?
Reservoir?
Is it where home?
Oh, no, not that
No.
one. It's in here.
So there's one in there that's supposed to be like to make like a house out of. So that's the one that best fit that I'm going with. But we all have our own opinions about, like is this a parasite or is this? I believe the cicada was already dead, or this is like the what is it? The skin of it. So like it's just the what is it? Nukegara, Nihongo de yuto. And it started building its house inside of it. So, okay, here's another one. So we've got a sea cucumber and a fish. So some of you guys may know this.
Symbiono? No. Maybe has habitat?
Is the fish happy there or not happy?
So this is a pearlfish. It lives in the butt of a sea cucumber. Yeah, that's what's going on. So like, let's try to look for the one that sort of kind of fits that definition.
This one.
There's another word in there that kind of best describes these two relationships.
Co-roost.
Yeah, these were all like ontologies taken from GloBi too. So like I just printed out all the possible things that you could associate with your observations.
Co-occurs with ectoparasite of dispersal vector of preyed upon,
kills endoparasite. You could kind of call it an endoparasite.
I think there's one that's like a commensalist. There's a commensalist one in there. So I guess nobody grabbed it.
Okay, apparently.
So we have no winners here. But
I mean, I have like half the cards.
wait, I think the next one's more interesting. Okay, here we go. We have a limpet and some worms.
I'm just gonna find the funniest one.
Parasite something? So this
But I think like sometimes from photos, it's hard to see if they're beneficial or not beneficial too. So that's something to think about.
Wait, this one? What's the difference?
What are the two cards? What's the difference?
Has parasitoid and parasitoid of. So I guess parasitoid of is from the perspective of the parasite, and has parasitoid is perspective of the host. So
Yeah, we'll go over some of that stuff too, like when we
Yeah,
get
this
into the
one I
machine
would guess.
readability of some of these things. No? Okay. Maybe not this one then. Okay, here, this is an easy one. Cowbirds.
I don't know. I know what it is.
If anyone wants to, for those of you who just walked in, we're playing a game. You can win a prize if you pick the right interaction.
Like co-roost.
Oh,
Yeah, there's co-roosts with
It's really more like
it. There's
Remember what's going on in this photo with the,
I actually
kleptoparasite and klepto something.
kleptoparasites. These are the kleptoparasite pairs.
It's,
there's one here too. Are you not gonna pick a card?
No, leave it.
Are you sure? Yeah, I still have four
I
keychains.
technically have the cards, but just because I have like 80% of the cards.
No? I think like you kind of said like the closer one with the
The roost.
co-roosts.
Co-roost.
This is the
Anyone, I
co-roost.
mean, this is someone also said said one of the possible answers too.
Kleptoparasite.
Kleptoparasitoid.
You know what it is, someone just said it, who has the card for it.
yeah.
He told it, I think.
Okay.
You can just keep it.
Okay, who has the kleptoparasite card?
This guy.
All right, congratulations. Yeah, so cowbirds lay their eggs and have other people raise them for them, so there you go. All right. Yay, winner. So this is actually one I took on Canvas. And this one should be easy too.
Pollinate?
That's one, that's possibly one.
Has flowers visited by.
Look very closely at this bumblebee,
Visits flowers out?
that too.
Have parasites on it. Those lights.
Praise on.
Not praise on, look very closely at this bumblebee, there is something wrong with it.
Dispersal?
The bee.
I mean, it has some weird bumps on it. I don't know what. Kleptoparasite?
Pathogen?
Not pathogen, you're getting closer though.
Parasitoid has parasitoid?
No, close, but we're going for something similar, but
The kleptoparasite is not right?
no.
Has vector?
The host.
A levopath
No, not that either.
of?
There should just be a simple one in there.
Epiphyte host?
No, not epiphyte either.
I have no idea what these things mean.
I mean, it is pollinating, but
Ecologically co-occurs with,
Wait, she said simple, so there's the simple pollinates, but
with
it's
pollinated
with,
by
we're
it.
going with the B right now.
The bee
Like
has-
take
Has pathogen?
Has pathogens?
has parasite. It's the mites, guys. The mites.
Has parasitoid.
It should be a has parasite.
Parasite.
The parasitoid is inside, you know?
I did not know that.
Yeah, it's very subtle differences, and here's where we're kind of like discussing.
Ah,
All right,
it was
winner.
upside down.
Here you go.
Thank you.
Alright. So are we kind of getting the hang of what we're looking at here? Okay. Here's another fun one.
Is friend of friends. They work together.
Symbiote.
Co-occur? No. Or they work together. Where are they?
There are many still available.
Can you just be BFFs?
I mean, yeah. I don't think I have a has BFF cards. It's not on the official ontology list of interactions.
I guess they're mutual.
They visit for tea.
Stalked by.
They interact, they co-occur.
So we have co-occurs with so far,
There's ecologically co-occurs with.
ecologically co-occurs with,
There's mutualist of.
mutualist of.
Co-occurs with is already there.
I mean, at this point, I might as well just take the BFFs of.
Mine's still be.
Oh, here we go, yeah.
So what is the technical term for that?
I think it was like a mutualist. was
Yeah,
what
I didn't see it up there.
Yeah, I have it.
I'll take the BFF. So I think we should just continue on. So it kind of depends, doesn't it? So like if you say host, like this is just general, like as a joke. I don't know. My joke slide. But host, house, like it depends on your background, like what certain things could be. Then we kind of have to think about like, well, what are we studying, and then what are those interactions going on? Because we had a little discussion about is this a host of, or the parasite of, or like host by. So we're gonna get into what is GloBi. So this is a global biotic interactions. So that's what GloBi stands for. It's an indexing tool, so not quite like a querying system like Symbiota or Ecdysis, but you can upload your own interaction data here and make it accessible and viewable and also usable by other researchers. So this is like an indexing. So when I actually go to GloBi, which I will do in a bit, you can get like the specimen citations, and I'll also kind of show you how to do it in Bash as well. So I told you this was a Corona project. So we would sit around in a Zoom and start reading literature and reports, and then pull out the interaction data in those papers. So like this Seratina visited some flower, and then we would pull it out and put it in a Google Sheet. And this went on for like 6 months until we burnt out. So we're taking those human observations and also the natural history specimens, like right now. Thank you. And taking that unstructured data and making it reusable, unstructured, and understandable by people and by computers. So it looks like this giant spreadsheet here. And so where do we get some of these? iNaturalist. So through iNaturalist, if you don't know what this is, we do have an ongoing BioBlitz at Spinach and Tadwig. You can download this free app. This is our, we have a lagoon on campus. So like we use it for species monitoring on our lagoon. You can download the app, take pictures, and other people identify it. I found a giant African land snail yesterday, and I don't know what to do. Outside, yeah. So So the example I'm going to use here is Bombus bosnicenskii. So we have a healthy colony of these out in our lagoon. So yeah, we do like some collecting and observations of these guys and the plants that they interact with on campus. So we have a bee pollination team and also a phenology team of students who go out and make these observations. So essentially it's kidnapping grand larceny of bumblebees. And we're stealing their pollen to see what plants they are. We're using the bees as, like to get their pollen. So, and then to figure out like what plants they're visiting instead of us just like watching where they go. So we steal from them, and then there's another master's student who's out doing the DNA metabarcoding. And then this is the barcoding results or herb results from last year from Bombus bosnicenskii and the different plants they visited during what month at the lagoon. So this is sort of like what we're doing. There's also like a phenology study out there too, but right now like this is where we're at. So the biotic reactions, source, relationship, target. So our source here is the bee, and then target is the California poppy. And for this particular one, like we did examples of floral visitation. So some of the bees are just like resting, but we do make a note of like, are they nectar robbing? Are they collecting from what flowers, pollinates, visit, floral visits? But for this one, we were wondering like, what plants does this bee visit? So in GloBi, the network looks like this. It comes out to this giant relationship map. So based on, like the visits flowers, collecting flowers, pollen, whatnot, I actually got this interaction of the spider and the bee like a week before coming here. It's kind of cool. So this is from a different study we did on them visiting lupins on campus. So here we have the source taxon ID, the taxon name, so Bombus bosnicenskii, and the interaction ID from the OBO library. So OBO is the ontology database of those specific interactions. So when you give it that interaction ID, like this RO whatever, the interaction name itself is visiting, blooming, and then we have the taxon ID for the lupin. And it kind of, when you go to the relationship ontology, it's gonna look like this bottom area right here, like what is those properties? So like there is like a standardized language of these biotic interactions and ontologies. So, okay, so now that I've kind of like gone through the slide, we're gonna hop into this workshop now for the nitty-gritty. For those of you who did do like some setup or have never seen this page, that's okay. I do have online, like you can check it later, is this test interactions.csv. We will minimally use this Google Sheets thing just to like look at some of the downloads. Hopefully I can finish this in 2 hours or less. So let's start. So here we go. I'm also going to, oh wait, no, this isn't the screen I want to share.
BFFs again.
Right. Where did it? How would you split screen? Oh, okay. All right. So I've downloaded my interactions to my desktop here. So I downloaded that CSV. It's kind of large, so you might not be able to do it on the convention Wi-Fi. But I do have this interaction, this like data folder where I've put all my data. There are some other things in here that I've kind of pre-made to make things go faster in case like the download speed here didn't quite work. But we'll mostly stick with, I will mostly stick with using simple commands in Bash to show you how you can use it. And there are a few other like smaller packages that were not mentioned in the lesson that I need to make a note of to get like some of the interaction reviews, but I'll just show you those. So for now I'm on my desktop, so, oh God, how do I, can we all see this? Hopefully. Let me put this away. Here. So I'm gonna change my directory and go to my desktop. So CD means change your directory in your terminal. And it lets you move through, the files on your computer. So you can also use CD and then dot or CD on its own, and it'll take you to your home. So just in case we haven't gone there. So I'm gonna go into my desktop. When I use the command ls, it's gonna list all the things on my desktop. So I've got like my RDP, Microsoft files, and also like the stuff for this workshop. So those are all the files that I have currently on my desktop. I'm going to move into this data folder that I have, maybe should not have named data, but just to keep things simple, I'm just calling it my data folder. And when I list the things in my data folder, it's got all this stuff in it. So some of it I made last night. So we're going to go into, we're gonna first run the shell command for GloBi. So let me see where I was, sorry. Yeah, so we're gonna do a word count of the interactions CSV here, so. Let's see, where did it go? Yeah, there you go. So when I'm gonna open this here, so you don't need to open it. Where is it? Oh my God. So when I just open the interactions CSV, it's kind of gonna be a mess if it does open. So when you're working with large data sets, sometimes you only want to pull out the stuff that you need. So in this case, we're going to do a word count, and then we're going to pull out the Ixodes data set. So we are working with terrestrial parasite trackers, like a data set. And I guess this might not be open. Okay. But when I do open it, it's going to be a large spreadsheet. So I'm going to just start by doing a word count. This is an L, not a 1. And interactions.csv. And it's gonna think a while because this kind of takes a long time because it's a large file. And if you're following along and you're following with me, could I see a green or red sticky if you have one? No? Okay.
What is the minus L standing for?
It's for, it's a flag. So when we do word count, we're asking for, hold on, WC. What was the L flag again? Actually, I think I can.
It's for lines. Count.
Yeah, it's for the line count. Yeah. Oh no, I quit. Let's go there again. Yeah, so it's for the line count. I usually work on a Windows, but my computer died before I got here, so I got a new Mac. Sorry. And thank you. So yes, word count, and line count. So it shows us that we have about 1.5 million lines of data, and that's a lot of data, and it kind of takes some time. So I'm in data. So what we're gonna do is we're gonna pull out only the observations that we want, which in this case, we have a shell script in here, so to pull that data out. So I'm gonna use the command sh. So I do have this shell script, the GloBi Ixodes data, so sh. And I'm gonna do that. What? Do I have to force quit? Okay. No, I don't want it to.
Maybe it's still downloading the.
I don't know what it's doing. So GloBi. I'm also gonna use the tab to make it to auto. What? Has run out of application memory. I might have to get out of the Zoom then. Force quit this. Oh, it's the numbers, yes, that's right. Yeah, so that's how big that data set is. It's 49 gigabytes. So there we go. That should do it, yeah. So this will also take some time to pull out some of those observations. And we should get a smaller data set that's easier to work with when we do this shell script. It's linked on the workshop website. I fixed the link because before it was like going somewhere else, but there's the code for it. There's also a way to do this in R. How many of you guys use R on a daily basis? Okay, so you guys are okay. The rest of you guys don't use R? Or have not? Okay, so maybe we don't need to. I'm not gonna use multiple programming things today. We'll just stick with Bash. That's okay. And this will take some time. Are there any questions up until now? Feelings? Yes.
So are you, if we're able to, do you want us to follow along as you go, or?
If you want to, yeah, it's okay.
The file download's a bit slow.
Yeah, so that's why I realized that when I got here, I was like, oh, maybe this might be more of a demo than a workshop, but we do have the lesson and all the data available online, and I'll fix some of the links for getting some of the Bash packages, because there are a few things that I realized were not listed on the workshop website that were like script files. Yes.
Where can I find the workshop in?
Yes, so it's this here. So this is this URL up at the top, the Kristi Sarah GitHub Interaction Data Workshop. I also do have it listed directly from UCSB Carpentries.
Okay.
So if you look it up, you'll see the rest of the stuff that we are up to. Yeah, so this one is right here. And that link will take you straight to the workshop. It says register for this workshop, but that's okay. Close this now. So basically at the end, we're gonna do like a little review test, and then this is just a sample of some of them, but we'll get to a giant spreadsheet like this. It's still looking for all the exodes, so I'm actually gonna quit this, tonight. But basically, you'll end up with a spreadsheet or with a data set of just only the exodes observations. Oh, no, wait, that's too big. Sorry. That's not what I wanted. So here, theoretically, after you get, it finishes that shell script, you could do echo create headers, so this would create the header. So actually, I'm just gonna copy this here. Because I kind of pre-made my data set already, so. Oh wait, no, this I don't need, gosh. Sorry. So I'm also not in the right directory either, I just realized that since I got out of the CD data. So what I'm gonna do is head lines, and then oh wait, I'm already in my data folder, so interactions.csv. And then the little sign would push it into its own exodes dataset. CSV. Which I already have, so if I do this now, I think it's gonna, But basically, it would push out something that looks like this, CSV here, which is significantly less than 50 gigabytes. Once it shows up. Hello. Or did it not do it?
I think you.
Yeah, I may have rewritten it, so
Yeah.
we're just gonna ignore that when I did that. That's why I'm kind of scared. So. But later we do create at the end of this code, we do make a exodes data unique CSV, so unique observations. That one I believe I did not overwrite, so there it is. So it's gonna look like this. Yeah. And it'll give you all the information that was inputted in the metadata here. All right. You could also, like, from here, we can make, yeah, so this was, okay, yeah, so this is fine. We're not doing this in R, or importing a SQLite database, because I didn't say anything about that earlier. But basically, once you clean your data a little bit, you can get like a nice CSV of all of these tick interactions. Okay. Are we good? Green sticky or red sticky? Okay. Red sticky, what's wrong?
I can find your data.
So the data was on the workshop website. So this was from that one CSV file that was in the setup. And then we pulled out all of the, well, I pulled out all of the exodes, just that specific, oh, not this one. Those specific observations from this giant file.
Is the collaborative document URL correct now? I'm getting page not found, so
Really? Okay.
Oh, I see, it's supposed to.
Oops, we can't find that page. Oh,
Yeah, I found it.
okay.
I mean, you have to hack off the,
Data set. There's also like the optional scripts. So like, as I went through it, I realized like, oh my gosh, like I think these are missing. So that's a note. It's an issue I have to put in in the GitHub to fix that.
yeah, maybe if you could share that URL larger, because then
This
we
one?
then we can chat and share more information there. And I'm almost done downloading
Okay.
the file, and I can pass a USB around if people want to.
Yeah, so it's this one right here.
That URL actually wrote it down here, so.
What document is that?
So kristisarah.github.io/2024 -09 -05 spinach as in the organization -GloBi. And then thank you. I don't see anything in the Zoom so far. Oh yeah, the workshop URL. Thank you, Jason. This is the SQL that we're doing. All right, so now we're gonna go into GloBi itself. So I'm gonna open this. And, oh, whoops, that should have been open a new tab, I'm sorry. We're gonna use this computer. And once we get to GloBi, it's gonna look like this. So it does have other languages, it is also in Japanese. And you can look up different queries. So the first thing we're gonna do is, as an exercise for those of you who are following along, we're going to search for interactions that involve ticks. And then narrow it to mammal interactions. So that's gonna be our first exercise using this link here, which I will also throw into the chat. And in the collaborative document, which totally disappeared on me. Yeah, is it this one? I'm sorry. What?
That's in there now. So what kind of organisms do X organisms interact with according to? So let's go and look for some ticks. So these, mammals, was it? Just mammals in general? Oh, so it's just giving me this, okay. Scientific taxon names, okay, great. So, are we okay? No?
Can I see
I-
the collaboration document, URL give.org?
I just finished downloading the full file. If anyone wants it, I can share.
If not, we can just kind of like watch me struggle through this too.
It says it's 200 megs on the page, but it's actually 2.2 gigs.
Yeah, so like that's a lot, that's okay. So when I look up anything to do with ticks, we've got, so on the interface, when we start looking up interactions, it's gonna look like this. So here we've got Martin, which has parasite, this Ixodes hexagonus that somebody has uploaded. And you can tell here that like we've got stuff verified by all this Wikidata, IDIS, GBIF. And also the metadata and the publisher as well. So it's a neat tool, at least for citations. You can also review and discuss as well. Who is on this website if you're following along? Green sticky? Okay, green stickies.
What was the keywords again?
I looked at the scientific name Ixodes. I don't know if I'm even pronouncing that correctly, I'm sorry.
My smartphone would probably hate on it.
Is that it? Oh, it was the other way around, see? I think I should have just kept it with this. All right. So, yes, see here. There's also an interaction browser, so when I go back to the top. So the other, when we first made this workshop, it was a Zoom workshop, so that exercise was like if we were in different Zoom groups. So let me go back to the data. I'm sorry. Not how to search. Where did that thing go? There it is. Yeah, so we can also get like a geolog. There's the thing. Here we go. So here, instead of Chelodia, I'm gonna pick it. And here we can drop down and get, where is it? Parasite Hub. I think this is the thing that needs to get bigger. Here we go. So we'll end up with this nice thing here. And then you can also see that relationship map on this side as well. So I know someone was like, whoa, earlier, but I think my screen is a bit too small to show
Oh, you're
this.
better. I'm doing it on a smartphone. This one's not a good idea.
And theoretically, and you could also pick it by geographic location as well. So here it's southern US, I would think there's more tick interactions here, but I guess not. So we can see that this is, I don't know what that is, Dama? Homo sapien, okay, so somebody documented a human getting a tick. But this is a powerful tool for you to visualize like those interactions and also see like your target and what it's interacting with and how. So hopefully it'll be useful for your work. So we've got people, Felis is a cat, yeah, Felis catus. Dog. So this relationship chart will show you your results of your query. All right, we're about like an hour in, so why don't we take like a quick 5-minute break? Yeah? Okay. Are there any questions up until now? Or comments? No? Okay. Let's go on.
I do have a question.
Yes.
In the next session, are we going to be using more of the data, sample data?
I'm gonna try to get it to work, yeah, because last night it was a bit, yeah.
I have it downloaded if we want
Yeah.
to follow along.
So we already did this part, yeah. So we're just gonna go into taxon of names and then try to see if this will go in and hopefully I'll finish on time. So it's mostly just trying to get those data. And then any suspicious names in GloBi perhaps. But we'll go over that. So let's take a quick 5-minute break. Also do have one more thing.
Is it USB-C? Is it probably not USB-C, right?
I downloaded it and I was like, oh, this is fine, and then I looked at the size and was like, maybe not. Like the point was to get like a larger data set and then kind of clean it up a little bit, but I'm kind of thinking as we did this lesson, like maybe that wasn't clear enough in the thing itself, so I'm thinking about all the issues I need to put in as I go.
You could, if you're creative, you could stream the contents of the data set and get a small bit.
Yeah, so like we were supposed to take the interactions, then make it into like a little text only thing. So like I already pre-made my thing, so
Maybe you can just.
when I ran it, I was like, this makes sense, but then like actually doing it, you're like, maybe not.
Maybe you can like
So-
start off with like a 100 with like 10 tick data.
Well, like when we check the review, it is like a sample of it, cause I tried running the whole thing last night and I was like, no. So like I do have a like a smaller sample that like pops out the review later, so.
I'm just trying to get to the page where you just have everything for the workshop, but
It's not reviewing text. Just go to interaction data workshop.
Okay.
So not the 06. I did not put 06 instead of 01. I did like an abridged version of this workshop. No, no. Oh, this is
Kristi.
Yeah, Kirsty, it's Kirsty. I-K-R-I. It should be spinach URL.
So I went on to the.
Well, okay,
My.
yeah, okay, wait a minute. So the workshop website is this here, so right now we're on this thing, but It's a, yeah, interaction data workshop. Are you connected to the Wi-Fi?
Yes.
So this, until the date.
Why would your head didn't work, so it'd be -1, -0. Cause you get the first line out. Or -N1.
Oh, we wanted the, yeah. I did the run through it and I was like, oh, it's okay. Going through like, and you're actually like talking to people.
Yeah, no, it's hard, it's scary.
Also, this is like a lesson we wrote in 2019-20, and then we decided like, let's kind of do it and just
Sure.
see everyone's response and then already update it and push it into the things. So,
With the word back.
oh, right, yeah.
I thought you was the other.
Yeah, sure. So Katja and your like, why are you complaining? No.
You know, neither of them is on the.
No.
Okay.
But it
I know
is,
it's not
it is reacting to get
No. And then also like, I had an undergrad student when we first made this lesson,
Okay.
and then he was supposed to come to finish, but his passport didn't come in time.
Oh,
Yeah.
that's.
So that's why I'm kind of like checking the Zoom in, so I am moderator and host at the same time, so.
How long have you been at UC?
Since undergrad or since as a staff member,
That's that.
5 years, so that's my 5th year. But even like 78 workshops later, it's always like,
Yeah, so you're more on the like the data side.
yeah, so.
I know you wrote all about more of the infrastructure. We did a couple papers on this. I'm sure he's.
It's like more domain specific knowledge, and I'm like, I don't know if I'm that person either.
Great. So, the work we did together with all of his abstract and that was.
I'm not a taxonomist, like I'm not one either. I may know a little bit more, but. So let's wait for some people to come back a little bit, and we'll see if the data's gotten any better or worse. It is a large data set, so I apologize. I'm taking notes as I go along about how we should improve this lesson, and I will take notes from the audience as well. Still not working on the URL here?
I tried changing the keyboard to US,
Oh.
but it didn't work.
Really? Yeah. Oh wait, no, it's persistent, so. Was it in general language?
I
Maybe we could start
My
with
first
GitHub.
suspicion was the event. I shouldn't have that. Yeah, I do.
Thank you. Okay, sorry about that, some technical stuff going on. Are we ready to kind of look at this? Okay, so this is the visual the visual pane. So I like this one more than the other one, but who likes this too? So hopefully this other, like the interactions, like will show you a better, you can also save these too, I think. But hopefully this is a nice tool for you guys to visually explore that interaction data with the, at least for this one it's ticks. I don't know what each of you guys are researching, but hopefully this will be a useful tool for you. I'm going to go back here with the key points. And then searching for interactions. Okay, so we did searching for interactions. How many of you were able to get onto this page and find interactions? Green sticky, red sticky, good, okay. No? Oh, so where did you get stuck?
The Wi-Fi is actually working.
It is, yeah, so I'm thinking like maybe this is more of a demo now instead of a workshop, I'm trying. But
You're good.
okay. Now I'll just go on to the next thing then. So working with data sources. So we found our data source and we're gonna locate some of these things. So in the sources, I'm gonna go to sources. Oh, okay. How do I make things bigger on the stack? Never mind. So like, hold on. Yeah, there we go. I don't know why I can't get off of that page, but here we go. Okay, so for sources, let's see here. Browse. No, that's not browse either. What? So working with data sources. So here's the indexes of all the data sets. So these were uploaded by various researchers. And just as a reminder, it's an index. So from here we can see reviews, GloBi checks, and the status of the data. So some of these are archived. Let's see here. What was I gonna go from here? So I've actually went ahead and kind of done some of this on my own. So I don't know if I know the Wi-Fi is kind of not great, but I did make some files and hopefully I can present that in a great way. But as you can see here, there's a lot of repositories of researchers who have uploaded their data into GloBi. So for this example, I'm going to pick out the data that I spoke about earlier when we were doing our COVID project. So I'm actually gonna close this. Great. Now I lost my page.
Maybe just a question. So you have color codes.
Hmm?
You had color codes in the previous page. Like it was red and green.
Mm-hmm.
And I don't know if you can maybe take 1 second and explain what is the red and what is the green and the.
So this is I believe this is like the review status. Let me see here. So there may be some results that are maybe missing or have not been completed yet, because there is a review process in GloBi. So the green ones are they're 100%. So so this is actually what we did. So this one is for our Big B database, and it goes over the data review and the methods that we use to pull out those data. So hopefully people can follow along later, if they wanted to look at our data set and the methods that we use to, the methods and the review of the species that we, whose interactions we got. So here it's this catalog of live thing. And you can also download the images as well. So you can check the reviews here. I believe it's that something may have been missing. So with this red one, there's nothing. So it's not, there is a review, but it hasn't been pushed into GloBi yet or was removed.
Ah, okay, so
Yeah.
okay, I understand.
Yeah. So there's like NAMs here. Or maybe like they started the process and didn't finish it. Yeah. And you can submit issues for suggestions or bugs. We like that. Okay. I'm gonna go back there. Oh. So when we do the review, it's an output of the data that was indexed. So this took a long time last night, so I don't recommend doing this here, but theoretically, and also like this part was, I realized after going through the code that like I didn't have anything about Elton or some of the other scripting tools that we use. So with GloBi, there are 3 packages that we use. When I clicked on that review earlier, like it said Elton, Preston, Nomer. So those are different tools that we use within the GloBi universe to get those taxon names and pull those records. And it does kind of take some time. Like I ran it and then I fell asleep and then woke up at like 2 a.m. and was like, oh my gosh. So so here, when you do run this review, or you'll get an overview of your data. So I'm gonna see if I can get this. So Control F and then CC. So this was an older data set that we used here. So review. And this was a review of biotic interactions within the invertebrate zoology collection at UCSB. So this lists our biotic interaction, the interaction networks that were created, and taxonomic alignments, as well as catalog names, ranks, and counts. So you can see how if you have like a larger data set, this may like conk out your computer. So I only have a sample today. But we do have like those alignment results in CSVs and TSVs, if you're into whichever one, from each of the different catalogs. So we have Discover Life, GBIF, IDIS, and also worms. Are we okay? Green sticky, red sticky? Okay.
So the review badge means that your data set went through quality control or I mean, formally, or is it also controlled by you?
It's also, it's not so much of a quality control, it's just the reviews are done by the data set only. And it's useful for people who are submitting data too, so, or like anyone who wants to know more about a particular data set. Okay. So at the end of doing your review, it's gonna like spit something like this out, and when I import a review sample here, so this took a long time, so I use this equal import data global biotic reactions at the end of that Preston and Elton code, and it will spit out like this spreadsheet here of where it's from, for here we have an ecdysis record, and also all the occurrence IDs and catalog numbers. So the catalog numbers being the little barcodes that we gave all these specimens at the Natural History Museum. So this would be the theoretical output at the end, which I realized like, oh no. Are we okay? So here. So yesterday, so that was okay, so within data, after I ran some of the Preston things, where is it? Oh no, it wasn't this either. But yeah, the code does take a while to load, so it may not be ideal, but I thought I would run out of time, but I think like that's sort of about it. If there are any questions or concerns or other. Comments? No? Yes?
What was the code doing?
So the code was, so there was extra, I realized later, but there's extra, so this is a tool called Elton that is made for GloBi review, but I realized like the explanation for that wasn't in the lesson, because I think it was sort of, at least within our group, it was implied that we all knew what this was, but now I'm like, oh no. So Elton is something that would interact with GloBi to pull those interactions from these data sources that were listed in the review here. So that's something I realized later.
So is it like removing duplicates or?
No, so like it lets you pull it in. So let me actually, like through your terminal. So. So we did find the data sources and then, what was it? But I kind of had to hop through some hoops to pull some of the stuff out. And then, yeah. But you would get like a CSV of your review, which I don't know where mine went, so. Here you go. This was the data cleaning with the exodes that we kind of did not get to. So, sorry about that. This is something else, so, okay. I think that's sort of about it for me, unfortunately, as far as I could go. This was an alpha lesson, so I'm hoping to get feedback from people so I can update this lesson and also fix a lot of the things that were going on so we can share this lesson for other people to share with their own institutions. So, yes.
So in the GloBi browser, the interactive browser with the
Yes.
graphs and everything, after you filtered for species, there's an access full data set. Is that the everything? But then the download CSV data sample, is that?
That was
That's
just a
already-
sample that that I made for this workshop.
No, there's a, like I've searched for Quercus fusiformis and oak.
Okay,
And when I click on download data sample, it seems to be just that species.
okay.
See that download data
Yeah.
sample there?
Oh, so it's not bringing down the other stuff too?
Scroll all the way to the top. And that download CSV data sample link down 2 inches, 1 inch. At
Oh,
the very
this
top of the
here,
table,
yeah.
yes. So is that already pre-filtered to that species? I mean, it seems to be at a glance,
Let's see here.
so maybe that's a way to start with a
Yeah.
smaller data set rather than
Maybe,
filtering.
yeah. So it gave us ooh,
I don't know if it's all the matches, but it's,
yeah. Trying to think of like a better way to. This is all fishes though, Sarcopter, no,
well,
yeah,
you had a fish.
I did,
mean,
yeah,
that's
I
what
did
you
have one. So maybe make this a more generalized one instead of, thank you. But yeah, I am accepting GitHub issues. Anything else? Yes,
I want to upload the data set to
Mhm.
GloBi. How can you do it?
okay. Let me go back to I think, or was it is an indexing, so like not we, but oh no. Did we get that? Oh, I don't think I got that far for this workshop because it was just like I'll get back to you on that. Because I only just designed this to be like a slow, soft intro to GloBi. Now I can't get out of this page. Oh yeah, contribute, yeah. So you publish it, you can do it through Zenodo. Do you know Zenodo? Yeah, so you can do it through Zenodo and then add it to the Global Biotic Interaction community. Yeah. And then you'll end up on the data sources page. Here. Yeah. Okay. Are there any questions in the Zoom? No? Okay. Well, thank you for coming, and I'm sorry it didn't really work out the way I wanted it to, but I've learned a lot. And I hope you learned about GloBi as well. But it's a work in progress, and Jorit, who is the developer, is one of the developers for GloBi. He is a software developer, not a taxonomist, so he's always looking for more domain experts. I am not a taxonomist either, so he's always looking for more domain experts to help have better ideas and better have this data open and usable for everybody else. So I hope this was a resource for you. Hello, yes, in the back.
Maybe, so you said you might do this workshop again.
Well, I'm gonna fix it before I do it again.
Would it be possible to publicize and do it again?
Yeah, so if we were to ever do this again, it would be under the Carpentries. And when it shows, so this is the Carpentries upcoming workshops. So there is a website for Carpentries in general. Some of these are online, so the UC system has a giant online workshop. It says UC, but you don't have to be a UC student. We will start offering workshops on Monday. So Introduction to Tidy Data. They made us all cough up a helper and instructor from each of our 11 institutions. So these are very large workshops. So we still have positions for, so these are all Zoom workshops. If you would like to join, you may register. You do not have to be a UC student. So it's Introduction to Tidy Data, Unix shell that we went over today, version control with GitHub if you've never used GitHub before, SQL databases, Python, or OpenRefine.
Sorry, how did you get to that?
So this is upcoming carpentry workshops. So if we were to ever do another workshop like this, it would show up on this page. And also, first, there are steps you can make your own carpentry workshops. Those would be under the carpentry I cannot spell today. Incubators, trees. And these are a little bit more niche or very awfully specific to certain people. So there's camera trapping data workshops. So that's one I know some people I know are writing or doing. So I think there are different steps to put lessons into the incubators. So for this particular workshop, we would make it into an incubators, and then it would have to be taught by 3 separate institutions before it's kicked into the actual data carpentries. And then we'll argue about it somewhat in a Slack channel about what carpentries does this specific workshop fall under. I vaguely said data carpentry. I know it's not a library carpentry workshop. But here, this is one we actually made, the reproducible publications with RStudio. So we use Quarto publications. There's also Introduction to Deep Learning. So there's different stages. They're ingesting very specific workshops. You can also look at, you can also review lessons too, or teach, or browse lessons repositories. What do we have in here? Julia, if anyone's into that, introduction to Docker and using containers. So you can make your own workshops and lessons as well, maybe better than, what we're kind of working on, and also like you could also join different repos to also like help develop those workshops too. So if you're interested, you can join this project, I believe. So if I go to GloBi's GitHub, we have a lot of things going on here. But yes,
I was just gonna ask, so I went to Data Carpentry, then upcoming workshops, UC was not listed, so I really.
so this is carpentries.org, not Data Carpentries.
Okay, got it,
Yeah.
perfect, thank you.
UC is one of them, and there are other universities that are offering them. So there might be one in-person workshop that's closer to you geographically too. So not just the United States. Japan has a very small community of carpentries, carpentry maintainers and instructors, but they are hoping to increase the number of people who use the carpentries. So yeah, this one's online too, so you can tell by what country they're from. They go to Japan. Yes.
What would be the like prerequisite like knowledge about?
Nothing.
Okay.
Like if we want to hear, I can show you like what we start when we start off with teaching R. I know this is like jumping into general carpentries, but that's okay. That may be a little.
The most advanced programming I did was on Excel.
No. Back here. Past workshops. Okay, so when we did introduction to data workshop materials, okay, with no programming. So we have a summary and setup. We don't, at this point, like I've been doing this for a while, I cannot expect the students to actually do the setup and download R. So we actually use Jupyter Hub instances where our students log in with their credentials to have something where we've already pre-made logins. For those, and of course there are some people who are like, well, I'm not a UC affiliate. And we've made Jupyter Hub instances also. We've been testing out instances in Olama. So that's the only time I've heard of using Olama was for using it to make Jupyter Hubs. But when we start introduction to R, it's literally like 3 + 5. And like this is what the interface looks like. So like the bar is very low. Like we don't want this to be intimidating to everyone. There are no stupid questions, but I give stupid answers sometimes. So it's very rudimentary. We try to, make this as self-entry for you as possible. I would recommend, so like the lessons are free online, so you could go and follow this yourself. It is mostly a teach and type model though, so like we would sit here, like there would be tables, we would be typing along. But they're available for you, so you can review them on your own time as well. So even if I'm not here and also, or just if the instructor isn't here, there may be YouTube videos online of different carpentry workshops, but mainly they do not record their lessons to encourage the teach and type with the instructor. So it really is like, this is how you use, like this is what the RStudio interface looks like. Is it even in here? Yeah, before we start? Is it before we start? Why learn R? What is R? This is what R looks like. We have our R scripts, our console terminal environment, files. I know I'm like doing R right now, but hold on. If I were to go in here, and actually, what I'm going to do is go back into data, and I have there it is, the GloBi example. So literally we would open a data set like this and tell you like, slight diversion, but we would have some scripts and lessons ready for you. And in this case with R, this is an R Markdown file. So it looks a little bit different from a normal R script. Like you've got these little chunks and YAML headers. YAML stands for
Yet another markup.
yet another markup language. Yeah, it's something dumb. Like yet another markup language. And you can make it look like source, and then like the visual one will push it out to make it look fancier. So you've got like this Jupyter Lab notebook style thing going on here in chunks. Our studio uses libraries, which are like packages in Python. You can see those packages here, down here in this thing, and then install whatever you want. Your files will show you, where your root directory is, like a normal file thing instead of like in your terminal, but you also do have a terminal here if you want to use it, consoles, and then when you create things in RStudio, I'm gonna run this just for, okay, I have all my libraries, and then I'm going to do this, run this. And when I run this current chunk, and it's gonna spit out whatever it is that you're running into this environmental thing on the side. So basically, we just start from like very basic concepts in R, so be as least intimidating as possible, if anyone was wondering. And same thing with Python. I do not have a Python script on this computer to show you. But yeah, that's me pitching the carpentry. So if you're interested in making your own lesson that's like domain specific to you, like go ahead and do it. Yes.
So I just wanted to maybe review and see if I understood what the main goal was. So I know
Yeah.
starting with a large data set was a big problem.
With
Not too
GloBi,
big, yeah.
and then cleaning it up and then seeing those interactions, and then we didn't have like a GloBi like tick review, so we used like an internal the invertebrate zoology collection from UCSB to get that sample, but yeah, then
So mostly it was to filter the data set,
Yes.
maybe count how many interactions
Yes.
there were, and then get that into a spreadsheet in Google
Yes.
Sheets. Okay.
so it would kind of look like this. Whoops. So this was my goal, was to get here, but and this is only this many interactions. This is only a sample of the total interactions because it totally calmed down my computer last night.
So was that an additional formula you added, or was that?
Yeah, this was, I went up and I import data from blah blah blah.org, and then this was the review sample CSV.
Okay.
Yeah, so this was the only part of the Google Sheets. But I can't expect everyone to have a Microsoft license, so try to make this as accessible as possible. We have the archives, so I could click on this and hopefully it will tell me, oh yeah, it'll download the thing. I'm not gonna do that. And it'll also say like the institution, the collection codes, IDs. I remember making all these little barcodes when I was a student. And then in ecdysis, you can see it here as well. So there it goes, Lepidoptera puridae. Yay. All right. Are there any more questions about GloBi or just the carpentries in general? No? Yes?
Are there any future plans to host portals like GBAP? They have hosted portals, so they can directly provide their data through API or something?
I don't think either side, I think like GBIF has their own workshops, like they have the data use club and they host their own workshops, but like we have not really like we have not worked with GBIF to make like workshops like this. And I think like there may be some, overlap, because they do their own thing as well. So like there's a booth over there and I'm sure someone could tell you more about their, they have open hours, and I believe yeah, they have the open drop-in hours and also like some of their workshops, I believe. And I don't know if those are free though, so.
I already talked about it with them.
Oh, and they said not really?
We can do it actually.
They can do it?
Technically, I mean, we can be I'm a manager of a data portal in South Korea, Quebec,
Yeah,
and we would like to provide our data through GBAP directly.
okay.
So if there is any chance to become a hosted portal,
Mhm.
GBAP, so they said, oh, it's technically okay, we can just talk about it later. But so in the same sense, I was wondering if we can have a chance to become a hosted portal for Globis. So because we collect
Yeah,
a lot
so
of like data?
Oh yeah, you can, so the way GloBi deals with data is it's an indexing. So you put your data in Zenodo and join like a certain Zenodo group and it shows up under sources here. So like this massive thing it's all indexed under Zenodo. So if you use Zenodo, then I think like it'll show up it'll be indexed in GloBi and people can find it if they go to this website, yes.
When you say quarter, like
Portal,
Portal.
okay.
yeah,
Data portal.
yeah.
Sorry, my English not so good.
No, you're fine. So these were all like through Zenodo.
So
Yes.
are those are both occurrence records as well as publications that include interactions?
Yes. So here is the sourced. When I click on it, it'll take me. And then there's also, you can discuss it, and we have the authors and whatnot. Ooh, whoops. So yes, you can find it.
I have just a couple of questions on this. So I managed to find a data set
Yeah,
and a paper,
uh-huh.
and then I thought maybe I would be able to find this paper on Zenodo,
No?
but I could not. So, which I
Oh.
think it could happen. But okay, are all the paper peer reviewed, or because in Zenodo you could have preprints?
You could have, yeah,
Yeah,
it
you
could,
could have.
yeah, I wouldn't be surprised if,
With
I
a
mean,
surprise. Okay, and then I probably need to, because the search engine of Zenodo is not optimal, so it might be
No.
that I did not find it, but it's there. So-
should be somewhere, yeah.
Okay, I understand. And one more, so the paper is available. I managed to find the paper, not on Zenodo, but we can find it.
Mhm.
But I do not see any such, the formatting that they have, it does not comply with what you showed here.
Ah,
So
okay.
it's probably someone else did the transformation. Could it be?
Yes,
Okay,
yeah, and you could also do that transformation yourself.
yeah, I understand. So the
Yeah.
paper may not have the appropriate source files
Mhm.
for GloBi, but probably someone made the transformation, maybe not the authors, but maybe someone else.
Yeah, so we do like have so like they are under the GloBi like GitHub when I like poke around here. So here was like, what is this? This one? And then here, supported by, okay, I'm gonna go to, yeah, so here it took me to the thing and the full identifiers and whatnot. So some of them are, and then some of them are not, it just kind of depends on like what they've thrown in Zenodo, but
Okay.
yeah. So here you could also like ask questions in the actual GitHub itself. So here's like the zip file for maybe this data set. Yeah. Zenodo is not my most not too knowledgeable in Zenodo,
But in
unfortunately.
a sense, sometimes you put the pointer to the publisher, so in
Yeah.
a sense, we don't necessarily
Yeah.
need to.
I mean, we have the DOIs sometimes.
Exactly, so in
Yeah.
a sense, yeah. Okay.
So like also in the review, sometimes like they'll list a little bit more information as well. The methods, results, yeah. Catalog of life. And also references from where they pulled this from. So, yes, thank you for coming. And I've learned a lot. I hope you have as well.
Thank you.
All right, I guess we finished 3 minutes early, but now I can put all my notes in. はい、これでありがとうございます。